Right. So I'm Kristin Branson, a senior group leader and head of computation and theory research at Janelia. And on behalf of Janelia, I'd like to welcome you all to this workshop on 4D cellular physiology reimagined, theory as a principal component. This workshop was co-organized by myself, James Fitzgerald and Anne Hermundstad, both group leaders in the computation and theory area at Janelia, Michael Reiser, a senior group leader in mechanistic cognitive neuroscience area at Janelia, Allison Scrow, a professor at Boston University, and Janine Stevens, a director of scientific programs at Janelia. So this workshop is the 8th in a series of planning workshops for a new research direction planned at Janelia. For those that aren't familiar with Janelia, to me, its most defining characteristic is its focus on collaboration between interdisciplinary groups, including theorists, tool developers, and experimentalists. Its model is to identify research areas that are at inflection points, or an injection of such collaborative concentrated effort, and in particular development of new technologies and resources, can hopefully have a sea change effect. We've identified 4D cellular physiology as such an area. The broad aim of 4D cellular physiology is connecting the fields of physiology with cellular and molecular biology. We want to understand the relatively large-scale phenomena studied in physiology, how organs and organisms are built, function, and maintained, at the mechanistic level employed in cellular and molecular biology, how cells function and communicate. These fields have been largely separate historically, but new technologies on the horizon will allow us to perform the types of experiments and collect the types of data necessary to hopefully connect these fields. How do we decide which types of experiments or technologies to prioritize? How do we turn the large amounts of data we can collect into understanding, insight, and principles? We believe that theory can help us answer these questions, and our goal in this workshop is to start this discussion. We hope to explore how the field as a whole can do this conceptually. What infrastructure would be needed to organize this field? How would an institute like Janelia do this? How can we better integrate theory and computation with experiments? Finally, before we start this workshop, I want to make one announcement. For this 4DCP research area, Janelia will be hiring 15 new group leaders, and there'll be a 10-week application period for the initial round of these hires that starts this week. So please look out for our new website and an email from us about this coming soon. All right, with that, I turn it over to Janine, I think.
Awesome. Thanks, Kristin. And thanks so much to the audience for joining us today. I will go ahead and link the agenda in the chat box shortly. But just to let you know, each session will have about 3 or 4 speakers, and each speaker has a 10-minute talk plus 5 minutes for audience questions after each talk. We really want to hear from all of you in the audience, so please write your questions at any time using the Q&A box that you see at the bottom of your screen. You are also welcome to use the chat box to share your thoughts, your big ideas, your comments, but please, if you have specific speaker related questions, add them into the Q&A box so that we can track them easily. Also, each session will conclude with a 20-minute forward-looking discussion amongst all of the panelists in that session. As a reminder, talks will be recorded and posted on the Janelia YouTube channel a couple of days after the close of the meeting. So without further ado, let's go ahead and get started with session 1. And to do that, I'm going to go ahead and introduce Jané Kondev, who is session chair. Jané from Brandeis.
Great. Thanks a lot, Janine. So why don't we just get into it? We're on a tight schedule. So our first speaker will be Vivek and Anne from Janelia. And why don't you guys go ahead?
Great. Thanks, Jané. Okay, both 4D cellular physiology and our current research area, mechanistic cognitive neuroscience, or MCN, are trying to bridge several different scales of behavior in complex biological systems. And both areas aim to use theory and computation together with molecular tools and imaging to build those bridges. In MCN, the goal is to bridge between flexible animal behavior and underlying neural mechanism. And so today, Vivek and I want to give you a high-level view of an ongoing collaborative project that's looking to do exactly that in fruit flies. And we hope that this gets across one way in which iterative theory experiment loops work here at Janelia, and shows how our tool builders, project teams, and core support teams enable us to make those loops faster and tighter. Now, fruit flies, like many other animals, use a diversity of different localizing sensory cues to navigate, including the sun, polarized light cues, terrestrial visual cues, and mechanosensory cues like the wind. And in any given setting, individual flies tend to maintain default headings with respect to cues within that setting, presumably to facilitate dispersal as a collective. And we know from ongoing work here and elsewhere that when orienting towards these cues leads to negative outcomes, for example, heat or pain, that flies learn to orient away from these cues. And so for many of these behaviors in which flies use past experience to flexibly redirect their actions away from some cues and towards others, we think that flies rely on internal representations of where they are and where they want to go. And here by internal representations, we mean that the circuitry in the fly's brain maintains information about the fly's changing spatial relationship to its surroundings. And so we're interested in understanding how those internal representations are constructed and used in the brain. And so today we'll first tell you about a flexible internal compass that keeps track of the fly's current heading or where it is. And then we'll tell you about a behavioral program or a policy that's tethered to this compass and that structures the fly's behavior relative to a goal heading or where it wants to go.
So in the lab, we study the neural underpinnings of visually guided navigation by putting tethered behaving flies in virtual reality environments under a 2-photon laser scanning microscope, which allows us to monitor brain activity. There are visual patterns, visual scenes on these LED screens around the fly, and we monitor the fly's intended movements. So for example, in a flying fly, we look for amplitude differences in its right and left wing beat, and based on that, infer that the fly, say, wants to turn left if its right wing beat is greater than its left wing beat. That's proportionately fed back into the visual display, and the visual display will show a pattern moving to the right to give a sense of closed loop control to the fly. Now, if we're doing a learning experiment, we add punishment into the mix. And so essentially, every time the fly is facing a particular orientation in this scene, we might ping it with an infrared laser targeted at its abdomen. This part I won't act out for you. So where in the brain would we actually image? And so the region that we've picked out as being of interest is one called the central complex. It's in the middle of the insect brain. It's shown in blue here. And it's one that's known to flexibly link different kinds of sensory inputs. So for example, here, input from the eye to motor commands to, let's say, the wings in this case. Thanks to an amazing body of work from a large number of labs and inside and outside Janelia, but particularly project teams and labs here at Janelia, we happen to know the morphology and connectivity of all the neurons in the fly central complex. And we have genetic tools to target each of these as well. So in this, almost all of them. In this particular case, I'm showing you in false color EM reconstructions of one subpopulation of neurons whose arbors basically carve up, partition this donut into wedges. And then we can use these genetic tools I mentioned to put in calcium indicators, genetically encoded calcium indicators into just this population and monitor them as the fly is moving. So in this video from Johannes Selig, a former postdoc in the lab, you can see the fly top left basically moving on a ball. This is in complete darkness. And simultaneously, you can see there's a bump of activity, a localized bump of activity that the population of neurons is organized into that's tracking, and this is again calcium, remember, so it's a proxy for neural activity. It's tracking the virtual orientations of the fly in complete darkness. How well? You can see that from the red and blue lines up here. Essentially, they track each other, the virtual orientation of the fly, it's the ball orientation and the bump orientation. So this is sort of the internal compass of the fly. So we call these guys compass neurons. And in darkness, this is something that you have as well. If you shut your eyes or the lights go out, it's not like you don't know where you are at all. You update it using vestibular cues from your ears, but in the fly's case, proprioceptive cues serve the same purpose. But of course, the compass is only as good as its ability to tether to the outside world as well. And so the question for us is, how does the fly in a brand new environment tether to the cues around it? In your case, you enter a room and maybe there's an AC blasting from one side, there's a door on the other. How do you get your bearings in this room? And so we know something about the sensory inputs coming into the compass system. They come in through these pathways that end up in ring neurons. So the red neuron that you see here is an example of a ring neuron in EM reconstructions here. And the yellow neuron here is a compass neuron. So each individual one of these ring neurons sends its outputs to all compass neurons, makes synapses with each of them, an example of which is shown here. And as a sum, they basically connect all to all. Now, you have these different kinds of cues coming into the compass system, visual, wind, and so on. And it turns out that when we analyze the anatomical links between these different populations of ring neurons and compass neurons, there's a hierarchy within them. So Hannah Haberkern in my lab and Marcella Norman in Anne's lab noticed that the numbers of synapses that these different classes of ring neurons make onto the compass neurons and the location of those synapses onto the arbors of the compass neurons suggest this hierarchy. So essentially, evolution's built-in structure into the circuit to potentially prioritize ones that give you a really good estimate of compass heading in a natural setting. For this talk, we're going to focus primarily on visual features and visual inputs. And so here's an example of two neurons that might be carrying visual input from the left and right eyes. If the fly happens to be oriented in a visual scene like this is the Janelia landscape building. Of course, most flies are inside, not outside, but nonetheless, the blue neuron might have a receptive field, might respond best to visual features in this part of the visual field, and the green neuron to visual features in this other part of the visual scene. And just like these two neurons, there's a whole another set of ring neurons which together tile the visual field around the fly, both in elevation, both in azimuth and in elevation. And so the question that arises then is, how does this mapping work from a new visual environment, let's say, onto the internal compass? And we think that's enabled through plasticity in the connections between the ring neurons and the compass neurons, and that the plasticity rule is such that the strength of those connections depends on the coactivity of particular visual feature detecting ring neurons and active compass neurons.
So to see how this could work, we can unfold this donut so that compass neurons are arranged vertically and ring neurons are arranged horizontally. And Vivek mentioned that all of the ring neurons talk to all of the compass neurons. And we know that these connections are inhibitory. And so we can represent the strength of that inhibition by the size of these circles that you see here. And now we can consider a simple visual scene with just a single feature, this blue bar here. And that feature will activate one of our ring neurons, shown here in blue and shown here in blue. And at the same time, there's a bump of activity that's maintained somewhere in the compass. And we're going to assume it's maintained by this compass neuron that's highlighted in white here and in white here. And now the coactivity between this compass neuron and the ring neuron will weaken the synapse between the two, which you can see by this shrinking circle here. And now if the fly turns, both the compass bump and the visual scene will move. And so the compass bump moving means that this compass neuron is now active, and the visual scene moving means that this ring neuron is now active. And so once again, the coactivity between these two neurons will weaken the synapse between them. And so if we play this logic all the way through, you can see that this plasticity essentially maps the features of the visual scene onto the compass and creates a self-consistent relationship between the visual inputs and the self-motion inputs. And this is indicated here by this weakened inhibition that you see along the diagonal.
So the question is this how the system actually works? And so Sung-Soo Kim, then a postdoc in the lab, tested this by using 2-photon imaging and tracking the calcium responses of these compass neurons while the fly was in a scene just like the one Anne described. And what I'd like you to notice here is that when the bar is moving clockwise, the bump is moving clockwise as well, and vice versa. And so essentially that's the relationship that's locked in. If we're right about this plasticity rule, what should be able to happen here is we should be able to build in a mapping that maybe even is inverted. And so this is what Sung-Soo tried to do. He optogenetically activated just a subpopulation of neurons, of compass neurons in particular parts of this donut, and simultaneously in open loop moved these visual stimuli around in the reverse direction. And so after a few minutes of this, he let the fly kind of do its thing. And so that's what you see here. And what you notice is that when the bar is moving clockwise, so when the fly is turning in a direction to move the bar clockwise, the bump's actually moving in the reverse direction. So this would be as if you were turning left and your brain told you you were turning right. And so now we're trying to do experiments that build on this work. Chuntao Dan, a senior scientist in the lab, is leading these experiments where we're now training flies with heat punishment associated with particular orientations in this world. So how is this flexible representation of the compass heading now used for downstream computations? To start with, he's doing behavioral experiments and tracking the heading of the fly as the fly orients in this environment.
So if we begin by looking at the behavior of individual flies before they've experienced any laser punishment, we can see that the behavior itself is highly structured. So here you're looking at the heading or the orientation of the fly over time, and you can see that there are periods where the fly maintains a constant heading. These are called fixations, and these are punctuated by abrupt turns or saccades that lead to changes in heading, shown here in purple. And so we can use statistical tools to analyze fixations and saccades and extract a candidate set of control parameters that capture the overall structure in the behavior. And we think that the relevant control parameters are a drift rate that controls how long flies tend to fixate at a particular heading, and a turn bias that controls which directions flies tend to turn when they saccade. And then we can ask how these control parameters should optimally be tuned as a function of the fly's heading. And then we can use these optimized control parameters to build a generative model of a model fly that we can then compare to the behavior of real flies. And so if we optimize the control parameters for maintaining a preferred or a goal heading over time, as flies are known to do, then it turns out that a smart strategy would be for flies to fixate longer at that goal heading relative to other headings. And this could be achieved by maintaining a low drift rate at this goal heading. And we would also expect flies to bias their turns toward that goal heading. So being more likely to turn clockwise when the goal heading is on the right and counterclockwise when the goal heading is on the left. And indeed, if we analyze the behavior of real flies after we've aligned their behavior to their preferred headings, we see the same structure emerge in the behavior, regardless of whether flies went through laser punishment or did not. And so the fact that we observe this structure in the behavior for both punished and unpunished flies suggests that this structure is built into the way that flies sample their surroundings, and that we can think of learning as the process of shifting this goal heading while preserving this entire structured relationship. And so for this to be true, for learning to shift an entire structured relationship, the fly's circuitry would need to ensure that the fly's actions, here fixations and saccades, remain structured relative to the fly's current heading relative to a goal, regardless of their absolute orientations. And so this now raises a new challenge. How does the brain ensure that behavior remains structured as the animal's goal is changing? And once again, thanks to insights that we're gaining from the electron microscopy connectome for the fly, we think we have some insights into the underlying circuit computations involved.
So we already told you about this heading bump. Well, it turns out that information is propagated from this donut-shaped structure to other structures, and we think the heading bump essentially gets combined and compared to goal representations in these downstream structures to determine what the fly should do next. So these structures we can anatomically subdivide based on the phase of the bump. So if you imagine that this particular donut represents 360 degrees of heading space, you can color in, if you assume the peak of the bump is at this point, you can color in the other phases as you go around the donut. Well, if we use the connectivity of neurons that actually connect these structures to other brain structures to propagate this coloring through, you may get something like this. So compass neurons, for example, take information from one of these wedges to the like colored wedge in this bridge-shaped structure. Other neurons, colored here in, let's say, white here, connect some particular part of the bridge-shaped structure to the fan-shaped structure. And so pink to pink to pink would be like a zero phase shift for a bump that's at zero at the pink location in the donut. Now, if you look at the bump then, this would be the current heading in the donut. This would be a copy of that heading, let's say, in the fan-shaped body. Why might you make a copy? Well, we think that's used to essentially associate different kinds of positive or negative experiences that the fly might have with its current heading, and to track that over time.
And we think that these associations are stored in the synapses of putative goal neurons that innervate whole layers of this fan-shaped structure. And so if we were to read off the strength of those synapses, we might see a profile that looks like this profile in blue that you see here. And now we think that the copy of the current heading bump could be used to modify this profile by, for example, weakening synapses at a particular heading if the fly is being punished, or by strengthening those same synapses at that same heading if the fly is being rewarded. And so in this way, the current heading could be used to build and update an internal representation of a goal heading. And now we'd like to understand how the circuitry ensures that behavior remains structured relative to this goal heading.
And we think that this behavioral structure is enforced by specific subpopulations of neurons. So in this figure, for example, what you can see is a neuron that has a phase shift relative to that location I was telling you earlier about. So the pink region in the bridge is now connected to a different region, say the orange or the purple. And so these different neurons have different phase shifts, in this case, plus or minus 90 degrees. So if you have a bump in the donut at this point, in the fan-shaped structure, you would now have it 90 degrees shifted relative to the earlier location. Importantly, these are neurons, this population of neurons go on to unilaterally project to premotor structures that we think drive clockwise and counterclockwise turns.
So this means that in principle, the flies could control the behavior, these turns, as a function of the current heading relative to the goal heading. And we think that that computation is carried out in these phase shifting neurons by multiplicatively combining their inputs. And you can roughly think of this as computing the overlap between this blue goal profile and this white phase shifted heading profile. And so here we've indicated that overlap by the height of this white bar down below. But of course, this overlap will depend on the relationship between the current and the goal heading. And so if we now keep the goal heading fixed, but we shift the current heading relative to the goal, you can see that indeed we sweep out this structured relationship at the output, where we have the strongest output when the goal heading is on the right and the weakest output when the goal heading is on the left. And now this should start to look familiar. This should remind you of the structured turn bias that we predicted earlier would drive more clockwise turns when the goal heading is on the right and more counterclockwise turns when the goal heading is on the left.
Finally, there is an additional neuron type, and this neuron type has a 180-degree phase shift relative to the other populations. And so what this we think is ideal for is actually controlling the duration of fixations. And so what the next picture we're trying to build here is one where we think the fly just needs to learn what its goal is, in this case represented by this dashed line, and move that around based on its experiences. And the rest of the stuff is built into the circuit, the structure of the circuit, to come for free, essentially structuring its behavior according to that one goal.
Okay, so stepping back out, we've told you a two-part story today. First, about how sensory and motor inputs are used to build and update an internal flexible compass representation. And second, how this compass representation might be used to guide flexible behavior. And in both cases, we think that the brain has evolved anatomical motifs that build in ethologically relevant structure. And within the constraints of those motifs, the circuit exhibits a great deal of flexibility. But this general principle manifests itself differently at the level of circuit algorithms in these two different cases. So in the first case, we think that the anatomy of the compass system enforces an ordering over different sensory modalities. And within the constraints of that ordering, the compass can flexibly tether to different sensory cues. In the second case, we think that the circuitry downstream of the compass ensures that the fly's behavior remains optimally structured relative to a goal heading, but that goal heading can be flexibly updated over time based on the fly's experience. And so together, this iterative theory experiment loop that we've told you about has enabled us to map out these algorithmic computations that we think are at play,
and have begun to allow us to ground these computations mechanistically in terms of the anatomy of the circuit, as well as maybe making targeted circuit perturbations, physiology experiments and behavioral experiments happen that are guided by the theories that are ongoing.
Which is nicely highlighted by these videos below. More broadly, this work relied on a variety of different theoretical, computational, and data-driven approaches to be able to connect across these different scales. And it could not have been done without the cutting-edge tools and bedrock of a number of different project teams and core support teams here at Janelia, including scientific computing, the Connectome annotation team, Fly Light, Fly EM, the Genie team, and Janelia experimental technology. And last but not least, this work benefited from contributions of different sorts from many colleagues here at Janelia, And so with that, we would love to open this up for questions, and I will stop sharing.
Great, thanks guys for staying on time, and that was really a wonderful talk. There was a couple of things that popped up in the Q&A. Let's see. So Navis was interested in sort of this kind of coupling that you guys talk about. He says the experiments remind you of a modified bicycle in which the coupling between handlebar and the front wheel is reversed. Bike goes left when handlebar is turned right, and vice versa. That would be a hard bike to ride. How is the fly able to learn the reverse coupling, but human can't? Maybe that's a little.
Can I jump in and take this?
Okay. So there is actually a paper and maybe part of a book that Martin Heisenberg wrote, which is about whether flies can ride a bicycle. That may be relevant here, but no. It is true that as adults, we struggle with riding bicycles that are wired up differently. Kids seem to manage it better. In the case of the flies, we're not saying that the fly navigates successfully with this inverted compass. All we were able to do is invert its compass. So the fly might be thoroughly confused and unable to navigate in this setting. So that's what I'd say there. So it's a very unnatural situation.
Mm-hmm. Yeah, sounds like it. Yeah, so Pankaj, I wonder if he could be unmuted because he had left a fairly long, I don't want to read the whole thing, question in the chat, which I think relates to kind of a bigger question of the role of theory as sort of more maybe as defined by physicists in these kinds of in this kind of work that you guys are describing. So Pankaj, can you sort of try to state your question a little more succinctly?
Am I talking? Yeah, okay.
Yes, you're talking.
Okay, yeah. I think all I was saying is that there's a level of theory assumed in the work already, starting with the attractors, Hyams-Polinsky's work, Hoffield's model, and I'm wondering if we're even trying to develop that kind of initial theory in 4D physiology and how you would think these lessons would transfer over, because I feel like there's so much understanding already assumed at the starting point, I guess is how I would put it.
Right. So this work did build on decades of theoretical work that had been developed to describe head direction systems, and we've relied on that theoretical work to gain traction in this system. But we've also relied on sort of normative perspectives of trying to understand how the system should be constructed to achieve different or certain goals. And so that's allowed us to sort of couple those normative approaches with these existing theoretical. I'd love to get into the discussion in terms of what we would need to do moving forward in 4D cellular physiology to lay the right foundation, and what we're primed to tackle now versus what sorts of frameworks would need to be developed in order to make the type of progress that we've been able to make in this system, whether that be new theory or new tools or a combination. I don't know, Vivek, if you want to add anything to that.
Looks like Boris, aka panelist GT, has his hand up. So if you want to ask your question, Boris.
Yeah, sure. I think you can hear me. Actually, I just have a technical question. So Anne explained how the coupling between the visual field and the heading should produce a radial shift of, I believe, inhibitory zone in that circle structure. I didn't notice bumps moving radially, or for that matter, any sort of radial resolution in the movie. So how are we doing on comparing theory and experiment?
We illustrated that movie with that sort of radial packing just to be able to see the different ring neurons. So really it's the individual connections between the ring neurons and compass neurons within a particular wedge that's being strengthened or weakened. So we weren't trying to, and I think Vivek wants to add to this, or you should.
Yes, I was just going to say, if what you mean is we haven't yet shown experimentally that we can basically manipulate and monitor the changing strength of connections between individual ring neurons and individual compass neurons in just the way that we described, you're absolutely right. We haven't yet done that. We don't think the changes are in fact structural. They could be temporary transient changes captured by something like a calcium conductance being modified or something. So it could be subcellular. And we don't have any clue yet as to exactly what that is. I mean, there are ideas around and some data, but not enough to nail it down. So that's still unknown, but the radial thing was more to just show you, these are the bunches of ring neurons and so on and so forth.
Okay, I just
We should
overinterpreted the anatomy of that particular picture.
Yeah, sorry, I didn't mean to interrupt you guys, but we should be moving on. Thank you so much, Vivek and Nan. And our next speaker is Aubrey, also from Janelia. So why don't you go ahead?
All right, can everyone see and hear this fine? Great. All right, so thank you to the organizers for inviting me to share my thoughts on the connection between theory and experiment. So like Anne and Vivek just spoke about, there's really almost this magical regime that exists at the intersection of theory and experiment. And I'd like to elaborate just a little bit on the experimental side in the era of big data. A specific example is these high resolution, large volume electron microscopy data sets that have been made possible by Sean Xu when he was in Harold Hess's lab here at Janelia. And to acquire these data sets, it really takes inputs from multiple fronts, and sometimes it's almost a literal armory just to get the data. So, it starts from the biologist asking questions to chemists targeting and labeling, the objects of interest, and microscopists and technologists coming in to be able to then see those labeled objects. And then, after this initial mountain is climbed and the data is obtained, you have to find a way to handle it and to extract that information and make sense of that information and answer those burning questions that sparked this whole thing in the first place. So the sweet spot here again is right at that intersection. But the tricky thing about that intersection is, each field speaks their own language of sorts. So we need to be able to translate in order to communicate to each other effectively when we're working across disciplines like this. And we also need the ability to scale and fill in gaps along the pipeline to make sure that things keep moving from one team to the next in an efficient and productive manner. And so what's really cool is that, here at Janelia, one of the strengths is that we really invest in this kind of science. So we have a mechanism for this, which is the project teams that help bring this all together. It helps fill those gaps. It helps with the translation and the communication, but it's not just here to coordinate. It's also there to feed energy into those loops, into those iterations. And we've got a really successful model of building on things like FlyEM and Flylight, Mouselight, Genie, and COSM, the cellular organelle segmentation and EM. And right now, it's a really awesome opportunity. The time is right for additional input and collaborations here. So I'll tell you a little more now about my own experience in this space working with the COSM project team. So what I'm presenting here is just kind of a vignette of sorts, and is really just this awesome collaborative and collective work of a really talented group of scientists. And I'm just here as a spokesperson for what we've done and where we hope to go. So in the case of COSM, things started after the data had been collected. The data has been aligned. It has to be thickness corrected, contrast corrected, tagged with the correct metadata. And then after that, skilled annotators can come in and densely annotate every voxel in small regions to identify all the organelles, the cytoskeletal elements. So really we're trying to label as much as we can, to learn as much as we can, to then ask exciting biology questions. We then take that training data and feed it into machine learning networks. And then ultimately, to provide segmentations for whole cells. So this is when you start to get to the exciting stuff, from little snippets of manually annotated organelles to a whole data set full of all the information that you could ever hope for. But then it's not the end at that point, right? Because you still need to make sense of it. And I think we haven't even scratched the tip of the iceberg there quite yet. data sets are more than just a pretty picture though. We can get functional information out of them, and we can also use them as a tool. So some of these data sets, correlative light and electron microscopy is really popular and effective, and it allows us to pair protein specificity with all of the awesome high resolution and global contrast of electron microscopy. But one of the hard parts of that is it's really hard to align those data sets. They're two completely different modes of imaging. There's usually been a lot of sample preparation that has gone on in between steps, and they're just hugely different scales. So we found that we can use these predictions of organelles to then automatically align these data sets and take what used to be days of manual effort and reduce that down to a few clicks and maybe a couple hours. And then, once we have all this information, these segmentations can then start to answer questions, right? So step 0 is to label everything, and then we can start to find ways to measure them. And I think that this is where a lot of the theory comes in. It gets really exciting, and how do we make sense of this? So, just some of the simple stuff can even be what are the organelle numbers, the size, the morphology, what's the different contacts between different organelles, all this information is just sitting in the electron microscopy data. So then ultimately with COSM, we made all this data and software and code open and available through the data portal Open Organelle in collaboration with the FibSim technology group, Sean Shu and Song and Harold. Hess Lab as well. So it's all available on Open Organelle. You can access it through Fiji, get the code and software through GitHub. I think this is also a really important part of this iteration, is making these tools really accessible for the community and users. So we developed this pipeline to handle these data sets, and it was all on single cells on a cover slip. And now we're excited to start applying what we've learned and continue developing this for cells in a more interesting environment, so in tissue, rather than just on glass cover slips. And over the last 8 months, there's been a lot of incredibly exciting discussions about 40CP and Janelia, including the starts of this discussion. And it's just this really cool opportunity to be in this new world of tissue. And one of the underlying themes that I guess at least I've noticed is that it really has to be an interdisciplinary effort. And another underlying theme is that big data is the way of the future. And with bigger data, comes more information. So that's the exciting part, because we can learn more things. But I guess it's also a challenge, because with more information and bigger data, it brings on more computational challenges, but also exciting new biological questions that can be answered. So if we bring it all back to this loop between theory and experiment, it's this awesome collaborative way to do science. And at every one of these steps, there's opportunities throughout the pipeline to have that mini iteration, like Vivek and Anne were talking about, that you can tightly interact with the chemists and the biologists, or the chemists and the hardware developers, and all along the ways. And I think, the project teams really sit in the middle of that and enable those effective, communications, and all the while, this tight integration between these groups is how we get to answer the questions, which is the fun part. But importantly, and maybe I think most importantly, it also tells us how we know which questions to ask. Because sometimes starting with a question is great, and then you learn something, but then the next question to ask, and the cycle starts all over, and you keep learning more and more. And so with that, thank you all, and you can have some questions.
Great, thanks a lot. We have some even extra time. So let's see, I'm monitoring things, but maybe I'll just kick things off, because I was struck by something you said during your talk, which is that, you do have all this information, right? All this detailed information, and there, of course, you can use it to answer biological questions. And I was just thinking, in your group, how do you think about sort of using this information maybe to kind of come up with theory in the sense of like a more coarse-grained description? I mean, presumably that also has to start with the biological question. So anyway, I was just wondering if you had what the kind of experiences thus far are going from this very detailed information to sort of a more, very fine-grained information to kind of a more coarse-grained understanding of cell physiology, I guess, given what you're looking at.
Yeah, I think what's exciting with this new direction is this ability to cross scales, and with that is a lot of information to be obtained, right? So we can find out that this particular cell in this area just so happens to have a lot of mitochondria huddled together that are really fragmented and have really dense cristae, and it tells us all these bits of information about the metabolism, and then we can take that information and see where that cell is located within that tissue, or even within that organ, or where that, what the function of that organ is, and really start to like bridge across those scales to really understand, how something inside of one cell, how the organelles distributed in one cell can really start to, relay information into how the whole organ is operating. And so, yeah, I think that along the way, it's going to take just lots of conversations with people, right? Because it's going to take someone understanding the metabolism at the mitochondria level, and the function at the organ level, and which is exactly where this like loop of talking with everyone really comes into play.
Yeah, and I guess kind of the other flip side of that question is, since you mentioned the loop, is once you've developed some idea, at kind of the tissue scale, can you sort of then, or some theory, can you then how do you go back then and sort of test predictions of such a theory? I mean, how does that happen in your in your group?
Yeah, so I mean, to date, the group I've been working with, COSM, has been trying to just develop a pipeline for the analysis, right? But I think now, we can look forward and see just exactly that, like how can we iterate on things and start asking questions with people. And I think that, yeah, I mean, it's hard with big data because you can't just go back to the microscope and take, a data set that takes months to acquire every time you think of a new idea. But I think that, with all of these different computational and theoretical aspects, we can start to guide the questions. And then, with involving the microscopist and the biologist at the same time, we can really start to ask more targeted questions that make sense for the system and for the tools that we have in hand. So it's almost about finding the questions that work with the tools, and developing you always want to develop new tools, but you don't want to take too big of a step and then not be able to answer the question because the tool was too far out of reach. So it's about like making those small steps all together forward. Yes.
Mm. And thank you. And in the Q&A, there was sort of, I think, a somewhat more technical question, which is, how do you make annotations that are robust for training, especially if you're looking at fine structures of organelles themselves? This was by Shotan, if I'm pronouncing it right.
Yeah. So we use a few different softwares, but these are, they're essentially painting the voxels and classifying what organelle or substructure they belong to. And it's a lot. This is again where that like communication between the biologists and the annotators are really important, just to make sure that things are correct. But there's always, I guess, variation between personal biases and such as well.
Great, and I think it was Genevieve first, and then Hannah after her had their hands up. So if you want to ask your questions.
So I was just wanted to go off of the last person's question and sort of thinking about like normally you have a perturbation in your experiment, and since it takes so much time and energy to collect these data sets, have you thought ahead about what type of perturbations would be, you want to focus on, or would be the easiest to collect, sort of the low-hanging fruit? And also just ways to account for inherent variability in the data that you're getting now.
Yeah, so the variability is a challenge in and of itself. But it's something that we hope to absorb by having more and more variable data, I guess, right? So the more variety you trade on, the more generalized you can become. But then, in terms of, finding the perturbations, like it's tricky, right? But I would argue that, like in some, like mouse behavior experiments, like you have to plan things out months, if not years in advance, right? To get all the right lines bred and trained, et cetera. And so this is not so different in a sense of, it's you can't just jump back to the confocal microscope and quickly, change that plasmid and look at that cell. But having those targeted questions beforehand make, testing those perturbations, I guess, easier.
Right. So I guess my question was more along the lines of, have you guys already started that planning, and what are you thinking of, or are you still in that phase?
Yeah, I'd say a little bit of both. So we're listening a lot to, what are the burning questions in the field, and trying to decide what makes sense for, with the tools at hand and what, we can develop. Yeah.
So sorry, Hannah, I think we need to move on. So maybe if you can ask a question in the Slack chat, that would be great. Just to stay on time. So next, thank you so much, Aubrey. And next we have Guadalupe from the Salk Institute. So if you want to, perfect. Thank you.
Yeah, so hello everyone. Thank you for being here, and thank you organizers for giving me this opportunity to present at the workshop. Today I wanted to give you one example of how to integrate models with theory or mathematical modeling. And in particular, this example comes from my work. In the last year, I've been working to develop a computational model of mitochondria, generating the 3D reconstruction from tomography, and generating the dynamics and simulating the dynamic of the key molecular components. And in particular, what you see here encapsulating the organelle is a button. I mean, we have been focusing presynaptic mitochondria. And I want to, kind of focus on how mathematical modeling can be useful to bridge between different scales. And I will share a lot of, ideas and things that I want to do in the future. So, life is intrinsically a dynamical phenomenon. It's a kind of defining features of life. And it's also a multi-scale phenomenon that spanned through a number of order of magnitudes in spatial scales that are also tightly coupled with different temporal scales. And I think, a key question in biology is how to bridge between these scales, how to integrate our knowledge at specific scale and go up or down. And this to me is tightly related to the question of how the system behavior arises from its interacting parts. And again, I mean, maybe thinking at the cellular level or organelle level, we have a complex network, a complex reaction network in specialized geometries. And let's think again about mitochondria. I'm here showing you, the internal structure in green, that form a number of compartments. And also, some of the key components are specifically localized in the membrane, where other components are diffusing in space as ATP and ATP molecules. So how to integrate all these to get system behavior? And I think the answer is in using mathematical tools. And as I mentioned, I've been developing a spatial model for mitochondria. We hand trace all the membranes and generate 3D reconstructions that are very high quality reconstruction. That is what we need to do not only simulation, but also the morphological characterization. We work hard on this and deeply characterize the morphology and to understand if morphology contains information. And here I'm showing you, we use tools from differential geometry, in particular here I'm showing the mean curvature and the Gaussian curvature, and we use these to extract the structural motif composition in these organelles. And we combine these with the dynamic and the dynamical rules of the key components and their interactions with random walk diffusion. As I mentioned, some of these molecules diffuse in space, and we integrated all these using MCell. This is HM-based reaction diffusion simulator. That again, without technicalities, I mean, this means that we are really modeling molecules that diffuse in space and interact. So in this way, we can explore we can explore the interaction between diffusion in this specialized geometry with the location and dynamic of the molecules. But one of the things that is key, that is missing in all this line, is that once the models are built and we explore things, I mean, we need to go back to the experiment and validate the models. And this is something that is missing, and some colleagues at UCSD, particularly Daniel Schmidt at Sync Lab, have been working hard to implement genetically encoded nanosensors to measure ATP, changes in ATP concentration in mitochondria. In particular, I'm showing, this is just one cell, and in particular region, we can see how when we inhibit glycolysis, the ATP concentration goes down inside the organelles. So what I'm trying to say is that electron tomography is very powerful to resolve all the internal structure, has very high spatial resolution, but lacks temporal resolution. Whereas, using fluorescent microscopy, we have lower spatial resolution, but we have temporal resolution. So we can use the model in a way to bridge between these two scales. And for the last minutes, I mean, I want to focus on three things that, what can be further developed in this pipeline on how to map form to function. And I separated this. The first part is related to automatic segmentation that, I know that at Janelia, there has been great efforts to do microconnectomics, but this, I think this more nanoconnectomic that is relative what Aubrey was saying is it's a bit different. I think these geometries are very convoluted, very complex. I'm kind of wondering if the same tools can be used for that. And also I want to highlight that what we need is very high quality measures for the quantitative to characterizing these geometries, but also for the simulations. And the other thing is about the second part has to do with the quantitative characterization of these morphologies. We work a lot with Blender, that is 3D computer graphics software, is open source and free. They're very powerful, has a Python API. So I think there are many good tools but these tools need to be integrated into. We are using Blender, so we think that Blender is the right platform to do this. And here I'm just showing us an example. I use a skeletonization tool from the SIGA library to generate the skeletons of the organelles, and I generate an add-on in Blender to do that. So this is a call for to a community to contribute in this direction. And the last part has to do with the models themselves. I mean, I'm kind of dreaming of having a multi-physics engine, again, something free and open source. And in particular, is based on, again, the experience that I have with this model. I'm showing you here some traces that are, results of the simulations that I did with MCell. I mean, in colors, the different colors mean different location of ANTs in different regions in the membrane. And I compare, I wrote that model and I had to write a different model that is the same model, but a different description using already differential equations, and I compare between them. And I was very surprised with the results. And I think in general, defining which is the right description level for model, the one is the Pellovin, is very important. And so having, if we have this tool where you can write your model and then simulate your tool and whatever, description you need, I think will be very powerful. In particular, this part has been done already by the EMSL team, and EMSL has been coupled to BioNetGen, so this is done. But I think we need more. In particular, what I'm interested in also is in integrate this with partial differential equations. But again, I think there are very good tools. I'm just talking about, bridging them together or putting them together. And in the last years, I will say I start to get interested in axons, and I'm interested in modeling electrodiffusion and the propagation and action potentials along the axons. And for that, we need partial differential equations. Recently actin rings have been shown also in axons, dynamic active rings. And if we want to model this, we also need partial differential equations. And to model membrane dynamics, we also need partial differential equations. But if we want to map all this to function, in particular to synaptic transmissions, we need to go back to model at the level of agent-based models, because we know that there are few proteins that are very specifically localized, and sometimes the co-localization and alignment between these proteins is key. So we need to go back to this level of description, and that's why I think we need hybrid models. So this is kind of a call to Janelia and the community to contribute in this direction. And I want to thank all the people that participated at different stages in this project, in particular Tom Bartle at Salk and Padmini Rangamani at UCSD. And with this, I am happy to take questions.
Great, thanks. Thanks a lot, Guadalupe. I think I'll kick things off again. I was just curious. So in the mitochondrial work, you said that you were able to, if I understood correctly, you were solving diffusion equations in the sort of geometry that was defined by the ENM, right? To get some idea about the changes of ATP concentration with time. And you said something that you found some surprising things. So I was curious if you could say a little bit more about that. And then if I may also, do you have ideas about how you might begin to learn what are the sort of important for the biology aspects of that very complicated geometry? Is there a way to kind of take all that complication and maybe summarize it or come up with an idea of what parts of that are really important, what are maybe spurious and not so important for function?
Yes, we're working on that, and particularly because we just finished a paper just focused on analyzing the morphology, and we do see difference in the different organelles and with different locations. So next for us is to understand what are the implications of these differences in ATP production. That is what we're interested. That is one thing. And related to diffusion, so we are using agent-based models. So it's not that we are solving diffusion equations. So it's more that we are setting the rules and we set the diffusion and this happens. In the end, in the back of agent-based, the diffusion equation is solved, but we are kind of sampling the solution of diffusion equation. Cool. and propagating and moving the particle with this.
Let me also turn to the Q&A. There was sort of a couple of interesting questions. One was, how do you ensure thermodynamic consistency while doing trying to do physics over multiple scales, right? How do you link different scales, I guess is the question.
So at the level of the models that we do, we can do, we can test, that, detailed model is satisfied, and more the multi-scale. Well, we are not. I mean, when if the model is correct, the level that we are modeling, that would be enough, I think. Yeah.
Let me see. There's also some, I think, plenty more technical questions from Stefan. It says one of the dream the data that would enable us to both train and validate automatic reconstruction of organelles such as mitochondria from medium high nanometer resolution such as FibSem would be in a comprehensive library of plausible morphologies. Do you have such a library for mitochondria? And if not, how many examples do we need to get to the point of having such a library?
That would be awesome. I mean, so we have only, we have reconstructed only 12 mitochondria, and with internal structure only 9. But definitely, I mean, I feel very uncomfortable doing statistic with low numbers. And so it will definitely interesting to look at more. And we have the tools. I mean, so we have the meshes, we can do these in an automatic way and, measure the motif composition or the surface area, etc. I mean, this is, again, I mean, I'm highlighting Blender because since it has a Python API, I mean, all these things can be done in a very easy way, and for many organelles, for many objects. So, yeah.
I mean, a follow-up question was actually, are you also trying to, and I imagine not, but do you have a generative model that could actually produce mitochondrial morphologies?
Yes, yeah, that's, well, that touch more the work related to membrane mechanics, right? But yeah, I personally, I'm not working on that, but in my team might be people developing this in the future, yeah. And my broader team, I mean, on the team that I belong to.
And I thought this was fun. I mean, the last question was, can you use your reconstructions to estimate the surface area of the inner membrane of the mitochondria? How big is it?
Well, I mean, please check out our publications. I mean, this is something that we can measure. We measure everything, the surface area of the Kristin membrane, the surface area of the inner boundary membrane. So the different kind of compartments in the surface. For the methods that we use to generate this, we have that. Yes, and I think we use that also to extract information. Yeah.
Cool. I think, thank you so much, Guadalupe. I think that is about it for the first part of our morning session, and we're going to go on a break now, and we will resume at 12:15 when Anne will take over as chair of the session, and our first speaker will be at that point Ned Wingreen. See you then.